last class on Tuesday, we were going through Punnett square. So we were starting to, kind of dip our toes into the water of how Punnett squares work. Okay, here we go. And so last time we had kind of finished up with this example. So where we took a homozygous dominant yellow seed pea plant, and we mated it with a homozygous recessive green seed plant. Okay? And so we came up with this Punnett square here, where we have our parents. We have the homozygous dominant on one side, the other parent, the homozygous recessive on the other side, and we performed the Punnett square. Okay, and what we ended up seeing is that all the offspring ended up yellow. Okay, and which is interesting. So each of these seeds turned out yellow, even though it has one copy of the yellow allele and one copy of the green allele. Okay, why is it that all of these offspring ended up becoming yellow? Why is that 100% of our offspring are yellow here? Right, because the yellow is dominant, right? So that big Y allele, the yellow, is the dominant allele. Okay, so even though this little Y allele is also present, this green allele is also present, okay, the dominant allele will always mask it. Okay, and so for that reason, they all end up becoming yellow, even though they still have a copy of that green allele present. Right. So perfect. Good job, Kimberly and Ryan. Good. Okay, so now we're going to take another example, and let's say we fertilize, or we have these heterozygous individuals self-fertilize. Okay, so let's go ahead and try out this Punnett square, and let's see what we come up with here. Okay, so let me go ahead and draw this out. Do some black here. Okay, so if this is our Punnett square, oops, something like that. Okay. All right. Okay, so if these guys are self-fertilizing, what should be the genotype of one parent across the top? What do we think? Not big Y, big Y. Remember, we're self-fertilizing these guys in the F1 generation. Okay. Right, big Y, little y. Good job, Angel. So should have big Y, little y. Okay. And then same thing over here, right? If they're self-fertilizing, if they're reproducing with themselves, the other parent is going to be itself. So it's also going to have a big Y, little y here. Okay. Okay, so now we'll go ahead and do our Punnett square here. So what should we end up with in this first box? Right, big Y, perfect. Okay. Then in this top right box, big Y, little y. Oh. Big Y, little y, perfect. And then this third box here, right? Big Y, little y, perfect. And then our last box here would be what? Little y, beautiful, good. Okay, so now what we need to do is we need to now analyze what our Punnett square means. So we'll take this from the genotype perspective first. Okay, so what is the probability of homozygous dominant offspring? Looking at our Punnett square, right, 25%. 25% or 1 out of 4, right? Because we have one of our 4 boxes, okay, having the homozygous dominant genotype. Perfect. What about the probability of heterozygous genotypes? 50%, right? So 50% because again, we have 2 out of our 4 boxes being heterozygous. Perfect. And then the probability of homozygous recessive would be what? Another 25%. Perfect. Yeah. So 25% down here. Okay. Perfect. So now the next thing we want to do is we want to be able to analyze the phenotypes. Okay. So what is the probability of having yellow offspring? Right, 75%. Right, it's going to be 75% because we have 1, 2, 3 boxes that all have the dominant yellow allele. So here they have the big Y over here, which is going to code for the dominant allele. This one has the big Y and the little y, but because the big Y is dominant, it's going to mask the recessive one. Okay, so we're going to see another yellow offspring here. Same thing over here. Okay, and then so what would then be the probability of having green offspring? Right, 25%. Okay, so the only way you're going to see green offspring is if you're looking at a homozygous recessive individual here. Okay, perfect. All right, good. So with that, when we do our Punnett square, we see exactly that same trend here. So we're going to see the dominant Y, the homozygous dominant allele, okay, here, heterozygous, which are also going to be yellow, and then one green seed allele here. And so that's exactly what we see. Okay, we see 75% having the yellow allele, and then 25% having the yellow phenotype, and 25% having the green phenotype. Perfect. All right, good. Go ahead and clear this here. Okay. So that's if we were going from the F1 to the F2 generations here. Perfect. So as we see here, that when we do our Punnett squares, okay, there's going to be an equal chance of all of our alleles, going into one of the offspring, right? So there's going to be an equal chance of this big Y allele ending up in our offspring, as there will be a little Y allele, okay? Specifically for the parents. And so what we end up seeing here is we see when we cross our heterozygotes, we see a genotype ratio of 1 to 2 to 1. Okay, so what that means here is that we're going to find one of our four chances of having a zygote that has the homozygous dominant allele, okay, the big Y, okay. We're gonna have a 2 in 4 chance of having a heterozygote, the big Y, little y, okay. And then we also have a 1 in 4 chance of seeing a green phenotype, which is the little y here. So we see a genotype ratio of 1 to 2 to 1. Okay? 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay? However, we have a different phenotypic ratio. Okay? So our phenotypic ratio is going to be 3 to 1, okay? Which tells us that we have 3 yellow individuals. Okay? The phenotype is going to be yellow seeds, okay, for 3 of our 4 offspring here. Okay? And then we have a 1 in 4 chance of having an offspring with a green phenotype. So in some cases, and specifically for when it comes to heterozygotes, the genotypic ratio and the phenotypic ratio may not necessarily be the same. Okay? And so when we're going through the Punnett squares, the things that you need to be able to do is be able to determine the possible genotypes, okay, and the genotypic ratios you may come up with, okay, and the possible phenotypes and the phenotypic ratios that they may show up in. So those are the 2 things you need to be comfortable with, okay? Being able to analyze it from the genotypic perspective, and then be able to analyze it from the phenotypic perspective. Okay. Is this making sense so far? If it makes sense, go ahead and throw a check mark or a star or something, or let me know. Okay. All right, perfect. Okay. So that's going to bring us to the law of segregation. Okay, and so before we get into the law of segregation itself, is basically when we're going through this process of meiosis, right? There's an equal chance, if meiosis happens correctly, okay, there's going to be an equal chance of all of our chromosomes separating into our daughter cells. Okay, so for example, the chromosome 1, for example, from mom and chromosome 1 from dad have an equal chance of separating into individual daughter cells. Okay? And so for that reason, that also tells us that the paired genes, okay, the genes within each homologous chromosome, have an equal chance of separating into one of the gametes. Okay? And so we have an equal chance of inheriting one gene or the other. Okay, again, just you're going to either inherit one or the other. Okay? And so that's going to be mainly seen through this process of meiosis 1 here. Okay? And so this is the reason that we know our Punnett squares actually work. Our Punnett squares, like I said on the previous slide, tells us that we have an equal chance of inheriting one allele versus the other. Okay? And the reason for that is because in meiosis, okay, all chromosomes and our genes have an equal chance of separating into one of the gametes here. Okay, so this tells us that our Punnett square actually does indeed work. Okay. So now, quick question here. So which of the following genotypes could result in the dominant phenotype? Okay, and there's 2 answers for this one. So we have homozygous dominant, homozygous recessive, and heterozygous. Which of these is going to result in the dominant phenotype? Yes, so A and C are both correct, right? So if we have a homozygous dominant individual and a heterozygous individual, they're going to have the exact same phenotype. Okay? They're going to look exactly the same from a physical perspective, right? Perfect. Okay, so one of the things that becomes important for us to be able to do as as biologists and as geneticists is we want to be able to identify what someone's genotype is, okay, in order to then figure out what their offspring are going to do. Okay? The problem with that is when we're physically looking at someone, we cannot determine what their genotype is, okay, necessarily, okay? Especially if they're exhibiting the dominant phenotype. Okay? So if they're expressing the dominant phenotype, we know that they can either be homozygous dominant or they can be heterozygous. Okay, but we have no way of physically being able to look at their DNA and tell us whether they're which one they are, right? And so that becomes important because again, we want to know the chance of our offspring developing a particular characteristic. Okay, so for example, let's say we're looking at some sheep. We know the black sheep or a black wool is dominant, right? But if we're looking at a black sheep, we don't know if it's homozygous dominant or heterozygous just by looking at it, right? We know the recessive one if it's white, right? Because if it's recessive, it has to be homozygous recessive. That's the only way we're going to see that recessive trait. Okay, but if we're looking at a dominant trait, we have no way of knowing right off the bat whether we're looking at a homozygous dominant individual or a heterozygous individual. Okay, so we want to try to figure out what the genotype actually is. Okay, and we're kind of following me so far. Yeah, okay. So what we're going to do in order to determine what a dominant expressing organism is, or what genotype that dominant expressing organism is we're going to do what's called a test cross. Okay? And so for a test cross, what we're going to do is we're going to take that dominant expressing organism, okay, and we're going to mate it with something that we know is homozygous recessive. Okay? And then we're going to look at the characteristics of the offspring. Okay? The offspring are then going to be able to tell us whether the parent was homozygous dominant or heterozygous. Okay? And so we're going to take a look at how to do that right now. Okay? So here we are looking at some sheep, right? So here we got a black sheep. Okay, here we've got a white sheep. We know the white sheep is homozygous recessive, so it's going to have a little b, little b. Okay, but here we have a black sheep. Okay, again, just by looking at it, we know it's the dominant phenotype, but we don't know if it's homozygous dominant or if it's heterozygous. Okay? So in order for us to determine what it is, what we're going to do is we're going to mate these two together, and we're going to try to look at the offspring. Okay? And when we look at the offspring, okay, the offspring are going to tell us whether this guy was homozygous dominant or heterozygous. Okay? And so here's what we're going to do. We're going to go ahead and do this Punnett square. Okay, we'll do one Punnett square on this side. Okay. Let me see if I can move this out of the way. Okay. All right, so we know one of the parents is going to be homozygous recessive, little b, little b. So we're going to go ahead and list that on one side here, little b, little b. Okay. But we don't know what the other parent is. Okay, he's either big B, or big B, little b. So what we're going to do is we're going to do two Punnett squares here, and we're going to try to figure out the chances of inheriting each particular trait. Okay, so let's assume he's big B, right? So let's say he's homozygous dominant. Okay, and so we're going to list his genotype across the top as well. Okay. Okay. And then what we'll do here, so that's going to be one Punnett square. I'm going to go ahead and draw out the other Punnett square on this side here too. Okay, so again, this is going to be the other possibility. Okay. So again, one of the offspring, or sorry, one of the parents is going to be big B, or sorry, little b, little b. Okay. And then in this case, we're going to assume he's heterozygous. Okay. So we're going to say big B, little b here. Okay, so let's start with this first. So again, these are two different scenarios. Okay, what happens if he's homozygous dominant, and what happens if he's heterozygous? Okay, so we're going to start with this first box on the left. And so in our in our first box here, what would our genotype be here? Right, it'd be big B, little b. Perfect. Big B, little b. Whoops, okay. What about the second box? What would we get? Right, big B, little b. Okay, and then this bottom left, big B, little b, perfect. And then the bottom right, big B, little b, perfect. Okay, so all of the offspring here have the big B, little b genotype. Okay, so they're all going to be heterozygous. Now, let's go ahead and kind of analyze this from a phenotypic perspective. Okay, so what are the chances that our offspring are black in this case here? Right, 100%. Right, because again, they all have the dominant allele here. Okay? So all of the offspring in this case would end up being black. Okay, if the dad is homozygous dominant. Okay, or if this parent is homozygous dominant. Okay, and then so as a result, we would have 0 white offspring. Okay, so all of our offspring would be black if this parent is homozygous dominant. Okay, oops. Okie dokie. So now let's do this Punnett square on the right. So here, this is the scenario if he's heterozygous. Okay, so if he's heterozygous, okay, what would this genotype here be? Right, big B, little b. Okay, what about in the top right box? Little b, little b. Okay, and then bottom left, big B, little b, perfect. And then in the bottom right, what would we get? Little b, perfect. Okay, so based off of our Punnett square here, what are the chances, or what's the probability of having black offspring? Right, 50%. Okay, and then what's the chance of having white offspring? Right, another 50%, right? So half of our offspring are going to be black and the other half are going to be white if this parent is heterozygous. Okay, so just by doing this Punnett square, we're going to be able to determine, or just by mating these two, okay, and looking at the Punnett square, we're going to be able to identify an unknown genotype. Okay, so if all the offspring are black, then we know the parent had to have been homozygous dominant. Okay, if we see a half and half mixture where half of our offspring are black and the other half are white, then we know this parent had to be heterozygous. Okay, and so that's how we're going to be able to identify, okay, what the genotype of a dominant expressing organism is. Okay, we're going to take that organism, we're going to mate it with something that we know is homozygous recessive, and then look at the offspring. Okay, and so that is what we see here, right? So if all the offspring are, sorry, if the parent is homozygous dominant, okay, we're going to see all 100% black offspring. Okay, if the parent is heterozygous, we're going to see that we're going to a half and half mixture. Okay, where half of the offspring are going to be black or expressing the dominant genotype or phenotype, and then the other half are going to be expressing the recessive phenotype. Okay, all right, this makes sense. Any questions on how to do a test cross and what a test cross tells us? Now, this is one thing that has tripped people up a bit in the past. Any questions? Okay. All righty, here's then I'll go and just clear this here. Okay, cool. Okay. So that's how we know the law of segregation is working here. Okay. There's another law that's going to be important specifically in metaphase 1 of meiosis, which is the law of independent assortment. Okay, so if you remember in metaphase 1, we have independent assortment where the paternal chromosomes, okay, or the paternal homologous chromosomes are going to line up on randomly across the metaphase plate with the maternal homologous chromosomes. Okay, and so it's going to assort randomly. They're going to sit on either side of the metaphase plate, and so none of the chromosomes are going to influence how another chromosome is going to be oriented along the metaphase plate. Some might be on one side, some might be on the other. It's going to be random. They're not going to influence how they all line up and how they assort. Okay. The other thing that's going to be important here is that the genes, which all kind of, if you kind of extend this, the genes also are not going to influence, okay, where the chromosomes are going to be going and how those alleles are going to end up getting separated, okay, during the process of meiosis. Okay. So the genes are not going to be influencing, where the chromosomes are going to be and how they're going to be assorted. And the chromosomes themselves are not going to influence each other, okay, on how the chromosomes are going to be separated here. Okay, everything is going to be completely random. Okay, so and everyone is independent of each other. All of our chromosomes are going to be independent. All of our genes are going to be independent here. Okay. So for this reason, there's an equal chance of all of our alleles getting there's an equal chance of all of our alleles, okay, occurring. Okay? We have an equal chance of inheriting any one of these alleles because, again, this whole thing is going to be random from beginning to end. Okay? The assortment of these alleles is going to be random. Okay. All right, so now that's going to bring us to a dihybrid cross. Okay, and so a dihybrid cross, okay, so in the past, in the last, lecture and a half so far, we've only been looking at monohybrid crosses where our parents have only been differing in one characteristic. Okay? In a dihybrid cross, we're going to be looking at two characteristics that are going to be different. Okay, but again, each of these characteristics is going to be independent of each other, but they are going to be inherited together. Okay, so for example, we might be looking at plants that differ in their seed color as well as in their seed texture. Okay? Seed color being either yellow or green, texture being either round or wrinkled. Okay, so each seed is going to have a specific color and a specific texture. Okay? So if we're looking at a homozygous dominant or a double homozygous dominant organism here, it's going to have big R, big R, big Y, big Y. Okay, telling us that we're going to have round, which is the dominant allele, okay? Round and yellow seeds. In the case of a double homozygous recessive individual, we're going to have little r, little y, little y, and that's going to tell us that we have wrinkled green seeds. Okay? So again, these are two independent characteristics that are going to be inherited together, but they're going to not influence each other. Okay? They're going to be kind of independent of each other here, but they're going to be inherited together. Okay? And so what's going to happen here is that each gamete is going to have one copy of each allele. Okay? They're going to have an R and either a big R and a big Y, okay, or a little R and a little Y. Okay, and so that's gonna we're gonna kind of see how that works in a second here. Okay, so let's say we wanted to mate a big R, big R, big Y seed with a little R, little R, little Y seed. Okay, and so what we end up with here is let me go ahead and just kind of write this out. So here we're mating individual with big R, big R, big Y with little r, little y, little y. Okay. And so to do this Punnett square, because we're dealing with two different alleles, okay, we cannot just use our normal 2x2 Punnett square. Okay, things get a lot more complicated here. So instead, what we're going to do is we're going to make a 4x4 Punnett square. Okay, so I'll kind of just draw this over here. Okay, so our Punnett square is actually going to look something like this. Oh, try that again. There we go. There's 1, 2, 3, and 4. Okay, so now the big R, big Y is going to be one parent. Okay. Little r, little y, that's going to be the other parent. Okay. So the way that we're going to organize our Punnett square here is we're going to list each allele, okay, across the Punnett square. So what we're going to do is we're going to take the big R and the big Y. Okay, that's going to be our first allele here. Big R, big Y. Okay. Then we're going to, let me do this in another color. Then we're going to do these, or sorry, I did that backwards. Then we're going to do this big R with this big Y. Okay, so we end up with another big R, sorry, big R, big Y. Okay. Then we're going to take the other one, the other R, okay, and interact it with this one. So then we end up with big R, big Y. Okay. And then the last one I'll do here in green. Okay. And so what we're going to do is we're going to take this big R and we're going to interact it with this big Y here. Okay. So we're going to basically come up with every combination that our R's and our Y's can have with one another. Okay. Yeah, so Mario came up with a little math, kind of comparison here. It's kind of like FOIL here. Okay, when we're working with equations in parentheses, exactly the same way. Okay? And then we're going to do the same thing with the other ones. Okay, with the little R's, the homozygous recessive ones. So again, we're going to have, oops, this little R, little Y. Okay, I'm going to list that over here. Little R, little Y. Okay, and then we're going to have this other little R, little Y. Okay, then I'll go in this box here. Okay, then the next one will be what happened here? Oh, what happened? Okay, here we go. Little R, little Y. Okay, and then the last one again would end up being little R, little Y. Okay, little R, little Y. Okay, so now that we've listed all of the possible alleles across the Punnett square, now we're going to do our Punnett square just like we normally would here. Okay? And so what we're going to end up coming up with is in this first box, we're going to have big R, little r, okay? Big R, little r, okay? Big Y, and little y. Okay. And you always want to try to keep everything consistent. Okay? If your R's are in front, keep your R's in front. If your Y's are in front, keep your Y's in front. But the dominant allele you always want to put in front of the recessive one. Okay. So then again, if you were to do this one, big R, little y, and then this third one, big R, big Y, little y, and this last one, it would again be big R, big Y, little y. Okay. And as you can probably kind of see going forward here, we're going to see the exact same genotypes across the board here. Okay. And so what we're going to end up with are what we call double heterozygotes, meaning that they're heterozygous in both of these traits, okay, both the round and the yellow alleles. Okay. And so if each of the offspring is double heterozygous, okay, then we know that the phenotype, okay, so if they're all big R, little r, big Y, little y, then we know they're all going to be round yellow seeds, okay, for this particular pea plant. Okay. And so this is what we would end up seeing over here. Okay. So again, you see your big R, big Y, big Y. You list them across one of the Punnett squares, little r, little y, list them across the other side. And then you end up with getting, let me kind of clear this here. You end up seeing all heterozygous offspring, okay? All carrying all expressing the round and yellow seed alleles. Okay? So this becomes our F1 generation. Okay. Any questions so far? You know, this is a little complicated here. Okay. You're just doing an extra step. Does this make sense? Okay, cool. Alrighty, so now what we're going to do is we're going to assume that we're going to take all of our F1 generation, okay, all of our double heterozygous organisms, and we're going to have them self-fertilize. Okay, so they're going to reproduce with themselves. Okay. And so this is what we come up with. Okay. So again, you're going to do your Punnett square just like you normally would. Okay. So here was the F1 generation, big Y, big Y, big R, big R, or R. And then once you bring these two together, again, you perform your Punnett square exactly the same. So this first, let me go ahead and bring out my marker again. Okay. So our first allele would be big Y, oops, big Y, big R. Okay. And so we get that over here. That's that guy. Okay. Then we have big Y, big Y, little r, okay, which we see here. Okay. Then we have our little y, big R, okay, which we see over here. And then little y, little r, which we see over here. Okay. So now we've got all different types of allele combinations across each side of the Punnett square. And then the other side would be exactly the same because, again, you're self-fertilizing here. Okay. And so then when you do the Punnett square, and you, again, put everything together, and then you have to be a little bit more meticulous about understanding what your Punnett square means here. Okay, so here we have a big Y, big Y, big R, big R. Okay, so that tells us we have yellow seeds, and they're going to be round. Okay. Here we have big Y, little y, so they're still going to be yellow because it's heterozygous for the yellow trait. Okay, big R, so it's still going to be round, so it's going to be yellow and round here. Okay, same thing over here, so we're going to have yellow and then big R, little r, so it's still going to be round again. And same thing over here, it's going to be heterozygous for both, so it's still going to be yellow and round, expressing both dominant phenotypes. Okay. Now, if we go down to this box, for example, here, okay, we see little y, big R, big R. Okay, so little y tells us we're looking at a green allele now, because now we're looking at homozygous recessive. Okay. And then round, because it still has the big R, big R. Okay. In this box over here, we have the big Y, so it still ends up being yellow, but we have little r, so we know it's wrinkled here. Okay, and so on and so forth. Okay. So this one is a little bit more complicated in terms of trying to understand and trying to kind of analyze what your Punnett square is actually telling you. Okay. And so, like we see over here, we're going to have 4 equally likely gametes. Okay. So big Y, big R, little y, big R, big Y, little r, little y, little r, okay, and so on and so forth. Okay. And 16 different genotype combinations, okay, as we see over here. Okay. We have 16 different combinations that we see just because of the fact that we have 2 different traits that we're looking at. Okay. And so what we end up seeing here when we cross 2 double heterozygotes is we end up seeing a 9:3 to 3:1 phenotypic ratio, okay, where we find 9 that have the dominant, or that are dominant for both traits, both yellow and round. Okay, so 9 individuals that are yellow and round. Okay, 3 that have a combination of the dominant allele and the recessive genotype for different characteristics. So we see 3 green and round. Okay, and then we also see 3 yellow and wrinkled. Okay, which we see over here, and then 1 green and wrinkled, one that expresses a double homozygous recessive genotype here. Okay. So like I said, these ones are a little bit more complicated because there's an extra step or a few extra steps here, and analyzing these Punnett squares are a little bit more complicated. Okay, so that is that, and I'll just kind of leave it there. Any questions on how to do these dihybrid crosses? Okay, cool. Looks like we have no questions here, so I'll just kind of leave it at that, and I'll go ahead and clear it. So that's the end of our Punnett squares when we're looking at the normal, simple dominance and simple recessiveness. Okay. So now we're going to go ahead and jump into 8.3 here. Oops, hold on. Let me, actually, give me 1 second here. Let's stop. Okay. Whoa. Okay, is everyone able to see the 8.3 PowerPoint? Yeah, okay, cool. All righty, so we're gonna look at 8.3 now. And so 8.3 is going to be some of the extensions of the laws of inheritance here. Okay. So this is gonna be chapter 8.3. And so before we get into that, quick question here. So which of the following is true about Mendelian genetics? Okay, so the genetics we just talked about in the last chapter. So we have A, 2 types of alleles exist for every gene. B, alleles maintain their integrity and do not blend in each generation. C, when a dominant allele is present, a recessive allele will be hidden and does not contribute to the phenotype. Or D, all of the above. Which one is true? Yes, I'm seeing some D's, and D is exactly right. Right, so all of these are going to be true. Right, we're going to have at least 2 types of alleles for every single gene. Okay, we're going to see that our alleles are going to maintain their integrity. So what we're going to see is that our genes are not going to be blending when we're looking at simple dominance and simple recessiveness. Okay, and then the C goes back to the law of dominance here, where a dominant allele will always match the recessive allele. So the recessive allele will not contribute to the phenotype. Okay, if the dominant allele is present. Perfect. All right, good. So that was simple dominance and simple recessiveness. That's what we've dealt with so far. And so for that, to kind of on that note, Mendel was correct in that there were some traits that were not going to be blending. Okay. However, we also know things are a little bit more complicated than that. Okay. We do actually see different types of inheritance patterns, and we're going to take a look at a few of these going forward here. Okay. So some of these are going to include incomplete dominance. Okay. We're going to see co-dominance. We're going to see multiple alleles for a particular characteristic, and then we're going to look at some sex-linked traits. Okay. So things do get a lot more complicated here. So we'll take a look at each of these, and we're going to start by taking a look at incomplete dominance. Okay. And so one of the things that Mendel had concluded was that when we're looking at an offspring's traits, the traits are not going to be a blend of the two. Okay. However, that's only true for some traits, okay, and some characteristics. There are certain characteristics and certain traits where we actually do see a blending of our parents' phenotypes here. Okay? And so that is what we call incomplete dominance. Okay? So incomplete dominance is where you're looking at one offspring, okay, or an offspring that's a heterozygote, okay, and it's actually going to be a blend, or its phenotype is going to be a blend between its 2 parents here. Okay? So for example, we have a red snapdragon plant over here. Okay? We have a white snapdragon plant over here. Okay? And if they mate with one another, okay, red and white is going to end up forming an offspring that is pink. Okay, so you actually blend the 2. Okay, so one allele appears, but it doesn't exclude the other one from also showing up. Okay, so it actually is an intermediate blend of the 2 parent phenotypes here. Okay. And so we're going to take a look at how to do a Punnett square for incomplete dominance. And so this is what it's going to look like here. Okay, so with incomplete dominance, we don't have any recessiveness, okay, because like I said, each allele is going to kind of show up in its own, kind of show up and blend together here. So all we have are homozygotes and heterozygotes. Okay, we don't have any dominance, we don't have any recessiveness. Okay, and so when we're drawing, or kind of when we're identifying our organisms, okay, here in the case of looking at a snapdragon, if we have a red snapdragon, we're going to give it the genotype CR. Okay, so one allele would be CR, for C for color, R being red. Okay, so if it's red, it's going to be CR. If it's white, it's going to be CW. Okay, so C again being color, W being the white. Okay, so CR is our red, CW is our white, and each one is our allele. Okay, so this is one allele, here's our second allele, and one allele, and then the second allele here. Okay, so if we were to mate a red snapdragon plant with a white one, okay, we're going to do our Punnett square just like we normally would. Okay, so we're going to list one parent, there's red one on one side, so we're going to have CR over here. Okay. we're going to have the white parent, CW, CW across this side. And then again, you do your Punnett square just like we would otherwise. Okay? So here we would have CR, CW, CR, CW, CR, CW, and CR, CW. Okay? Now, because the genotypes for the offspring are all CR, CW, it's going to be a mix between red and white. Okay? So because they're mixing, we're going to end up seeing that these end up becoming pink offspring, okay? Which is what we see down here. Okay? So that's how incomplete dominance will work. Okay? So now if we wanted to get a little bit more creative, okay, and we wanted to take our pink offspring and make them together here, okay? Again, we're into our Punnett square, again, just like we normally would. Okay? So again, we're going to have our pink offspring, CR, CW on one side, and another pink parent here, CR, CW, all across the top. Okay? And then again, you do your Punnett square just like we normally would. Okay? So here we have CR, CR. So we have CR, CR in this top left box. Here we have CR, CW. So that's going to be what we have in the second box. In this third box, again, CR, CW. So we're going to have another one in this third box. And then in this fourth one, we're going to have CW, CW. Okay? And again, that's if you're mating two heterozygotes, okay, or self-crossing a heterozygote, okay, our pink offspring. Okay? And so what we end up seeing here is we see that the red and the white flowers begin to reappear in this new generation. Okay? Initially, we were just dealing with pink offspring, okay, but now here we see the CR, CW, so that's going to indicate our red. Okay? Here we see, or sorry, CR, CR, that's going to indicate our red. Okay? Here we have CR, CW, and that's going to indicate our pinks. And then here we have the CW, CW, which is going to indicate our whites. Okay? So the red and the white flowers begin to reappear in this generation along with the pink flowers. Okay? And so here we end up seeing a similar genotypic ratio, okay, when we're crossing heterozygotes. We still see that 1:2:1 ratio where we have one homozygote, two heterozygotes, and then one homozygote, okay, again. Okay? So, and then we also see the same phenotypic ratio, okay, a 1:2:1. Okay, one red offspring, two pink offspring, and then one white offspring again. Okay. So in this case, the genotypic ratio and the phenotypic ratio are in fact the same. Okay, okay, so that's how incomplete dominance works here. All right, so what we're going to do is we're going to do a quick example here. Okay, so example here is in humans, hair texture exhibits incomplete dominance. Okay. The allele for curly hair, HC, okay, is incompletely dominant to the allele for straight hair, which is HS. Okay. Individuals that are heterozygotes have wavy hair. Okay. What would their genotype be? Okay. Would it be HCHC, HCHS, or HSHS, okay, for a heterozygous individual? Right, so it would be HCHS, right. So again, if they're heterozygous, they're going to have one copy of each allele, so they're going to be HCHS. Okay. All right, so now next question. So Ned Stark has straight hair, while Catelyn Stark has wavy hair. Okay, they have a baby. So let's go ahead and do this Punnett square. Okay, so you go ahead and do this here. Okay, boom. Perfect. Okay, so let's say we want to do Ned's genotype first. Okay, so if Ned has straight hair, I'll put him on the top. If Ned has straight hair, what is his genotype going to be? What do we think? Right, he's going to be HSHS. Good job, Jack. So HS HS. Perfect. Yeah, it's getting our Game of Thrones on here. Cool. HSHS. Perfect. Okay. Now, if Catelyn Stark has wavy hair, okay, so I'll go ahead and put her in, I'll do purple here. Okay, so I'll just put, oh, I don't like that at all, actually. Let's do that in pink. Okay, so I'll just put Cat for Catelyn here. Okay, so if she has wavy hair, what is her genotype going to be? Right, it's going to be HS, right? Or HS to HC, whichever way you want to do it. So we'll put HC, HS, because again, she's heterozygous here, okay, for wavy here. Okay. So now what would we find in this top left box? What would our genotype be for this offspring here? Right, it'd be HCHS, perfect. HCHS. Okay. What about the second box here? Top right. Right, HCHS. Perfect. Okay. How about this bottom left? HSHS. Perfect. Okay. And then the bottom right, what would we see? HSHS. Perfect. Good. Okay. So now we're not quite done yet, because now we need to analyze what our Punnett square is actually telling us here. Okay. So again, let's take these in the perspective of our genotypes first. Okay. So what is the likelihood, or what's the probability of a homozygous pair, or homozygous offspring? Sorry. Right, 50%, right? We're gonna have 50% chance of having a homozygous offspring, okay, because we see 2 of our 4 boxes having the same allele, okay, being homozygous here, HSHS, HSHS. So 2 of our 4 boxes have the same allele present, okay? So what's the probability of having an offspring that is heterozygous? Right, it's going to be another 50%. Good. Okay. So now we've looked at the genotype kind of perspectives here. And again, we're not going to be worrying about dominant or recessive, because in incomplete dominance, we don't have any dominance or recessiveness here. We just have homozygotes and we have heterozygotes. Okay. So now we're going to look at this from a phenotypic perspective. Okay. What is the chance of us having an offspring with, we'll say, curly hair? Right, Kimberly, it'd be 0%, right? Because in order to have curly hair, you'd have to have 2 copies of the HC allele. Okay, here we don't see anyone that is HC, so we're going to have 0% of our offspring having curly hair. Perfect. Okay. What's the probability of having offspring with we'll do straight hair? Right, 50%, exactly. It'll be 50%, okay, because again, we have 2 of our 4 boxes having the straight hair genotype. Okay, HSHS, HSHS, so that would give us straight hair. Okay. Now, what's the probability of having offspring with wavy hair? Right, it'd be another 50%, right? So again, the wavy hair would be the people who are heterozygous, whereas everywhere where we see HCHS. Okay, so we have 2 of our 4 boxes again containing the HCHS genotype. Okay, so that tells us that we have a 50% chance of having an offspring with wavy hair and a 50% chance of having offspring with straight hair. Okay, okay, cool. All right, so that is how we're going to analyze our Punnett square for this. Okay. Any questions on incomplete dominance? Does this make sense? Okay, cool. All right, again. So I'll go ahead and clear this here. Okay, so that was incomplete dominance. Okay. The next type of inheritance pattern we're going to see is called codominance. Okay. So codominance is a similar kind of thing to incomplete dominance, But instead of blending the two characteristics, instead each of the alleles will show up in its own glory. Okay, and so an example of this is what we see in blood types. Okay, so both of the alleles are going to be expressed at the exact same time. They're not going to blend, but they're both going to show up. Okay. So here's an example. So an example is when we're looking at blood types. Okay, so blood types are determined by genetics as well. Okay, just like many other things. Okay, so if you're a homozygote, okay, a homozygote is going to have a genotype, or let me backtrack. If you're a homozygote and you have type A blood, okay, your genotype is going to be IAIA. Okay? And so people who have type A blood will have the A protein on their red blood cells, okay, which gives them type A blood. Okay? Now, if someone's a homozygote for type B blood, okay, they're going to have a genotype of IB, and so they're going to have the B protein on their red blood cells. Okay? Now, here's where codominance actually comes into play here. Okay? If we are a heterozygote, okay, and we have, if we're heterozygote, we're going to have the type AB blood type, so our genotype is going to be IAIB. Okay? So because we have two different alleles, both of these alleles are going to show up. And this is where we see codominance. Okay? So what we're going to end up seeing is we're going to find the A protein and the B protein both present on the red blood cells. Okay? So this is where we specifically will see codominance starting to show up. Okay? So you start to see the A allele and the B allele both showing up in their own individual glories here. Okay? We're going to see both of them. Okay? All right. So codominance is specifically, again, for heterozygotes, and both of these alleles are going to show up like we see right here. Okay. That's our codominance. Okay, so now again, when you start to make these two, or make two individuals that are heterozygotes, so two people who have type AB blood, you're again going to see that same 1:2:1 genotypic ratio. Okay? So for example, if someone has type A blood, they're going to have the IAIB genotype. Okay? And if you make them with someone else who has type AB blood, okay, you're going to make them again. And now we can have offspring that have type A blood, if they have the IAIA. Okay? If they have IAIB, they're going to have the type AB blood, which we see in these two boxes here. Okay? Or if they're IB, they will end up with type B blood. Okay? So we still see that same 1:2:1 genotypic and phenotypic ratio. Okay? So one homozygote with type A blood, we have two heterozygotes, okay? And then one homozygote with type B blood. Okay? Same 1:2:1 genotypic ratio. Okay. All right, so here's an example here. Okay, so in some chickens, feather color is controlled by codominance. Okay, so when a black chicken, which has the allele FB, mates with a white chicken with an allele FW, okay, all of the offspring are covered in both black and white feathers. Okay, so that's how we know we have codominance. Both feathers are starting to show up. Okay, so they have the genotype FBFW. Okay, so a farmer mates two black and white chickens together. Okay, so we're going to go ahead and do this Punnett square. Okay, so here is our square here. Oh, we were doing so good with this box until I blew it right there. Okay, so so if we have two black and white chickens, okay, one chicken, what's going to be the genotype of one chicken? Or one of the parent chickens, I should say. What are we going to be listing here across the top? Not FBFB, because again, we're going to be mating two black and white chickens. Okay? So if you have a chicken that's black and white, what is its genotype going to be? FBFW, right. So we have a parent that is FBFW. Okay. FBFW. Okay. And then the other parent is also going to be black and white. So what's its genotype going to be? FBFW, perfect. FB, FW. Okay. So now when we do our Punnett square, what would we expect to have in this first top left box? What would our genotype be? Right, we'd have FB. Perfect. Okay. What about in this top right box? FBFW, perfect. Okay. In this bottom left, what would we have? FBFW, perfect. FBFW, cool. And then in this bottom right box, what would we end up with? FWFW, perfect. Okay. W, whoops. FW. Okay. So now again, we're going to go ahead and analyze our Punnett square. Okay. So we're going to start off with the probability of a homozygous offspring. What's the probability that we're going to have a homozygous offspring here? Right, 50%. Right, we're going to have a 50% chance of having a homozygous offspring because in one of our boxes, we're going to have the homozygous where we have the black feathers. The other one, we're going to have homozygous where we have the white feathers. Okay, so we have a 50% chance of having homozygous offspring. So 2 of our 4 boxes are homozygous. Okay, what's the probability? Probability of heterozygous offspring. Right, another 50%, right? Perfect. Okay, because again, we see our FBFW, those would be heterozygous and then make up 2 out of our 4 boxes. Perfect. Okay. So now, what's the probability that we're going to end up with offspring with black feathers? %, right? 25% here. 25%, because we see one of our 4 boxes has the FB allele or genotype. Okay, so FB, that's going to give us having black feathers. Okay. What about offspring with white feathers? Right, another 25%. Perfect. Whoops. There's 25% here. So FW, those are going to be our white feathered offspring. Okay. And then what's the probability of having offspring with black and white feathers? 50%, exactly. 50%, because again, we're looking at our FBFWs. 2 out of our 4 boxes have the FBFW genotype. Perfect. All right. And that is it for codominance. Pretty straightforward. It works pretty similarly to incomplete dominance. The only difference here is that we don't have any blending, but we do have, the 2 alleles beginning to show up. Okay. Okay. Any questions on codominance? Okay, cool. So go ahead and clear that. Okay. Okay, so now the next inheritance pattern that we're going to talk about is called multiple alleles. Oh, so Kimberly's question, so do humans have codominant traits? Yeah, so there are some traits that are codominant. Blood type is one of them. I'm trying to think of any others off the top of my head. Let me try to think. There are codominant traits. I'm having trouble thinking of something off the top of my head. But if it comes to me, I'll let you know. But there are codominant traits that do happen in humans. So I'll think and I'll let you know on that one. Brown eyes, kind of. but there's multiple. Actually talk about eye color later. So eyes are much more complicated than that. So we'll get to that in a second. Okay. So the next inheritance pattern is multiple alleles, and so when Mendel was doing his experiments, he was basically going with the assumption that there's only 2 alleles in a particular population. Okay, which was a gross oversimplification because there are usually going to be multiple alleles over the course of a population. So, for example, here we're looking at rabbits. Okay, if we're looking at their fur color, we're going to see some that are going to be gray. We're going to see some that are going to be tan. Some are going to be like a dark brown. Some are going to be white. There's going to be some that are black. So there's multiple different alleles that are all going to contribute to different characteristics. Okay? If we look at human hair color, okay, we're going to have people that have blonde hair, black hair, red hair, brown hair, okay, and so on and so forth. So there's going to be multiple alleles in a population, usually more than just 2. Okay? So even though each person has 2 alleles for a given gene, okay, because you're inheriting one from mom and one from dad, in the entire population, there's going to be more than just 2 alleles, okay, present. Okay? And so we call the most common genotype or the most common phenotype the wild type. Okay? Now, wild type doesn't necessarily mean that it's the dominant trait. Okay? All it means is that's what's the most common at that point in time. Okay? So a wild type is just the most common phenotype or genotype. Everything else, the other alleles here would be, or the other alleles or phenotypes or genotypes would be all considered either variants or mutants. Okay, so let's say for example here the brown color is the wild type. That's the most common in the population. Okay, then gray, tan, white, black, those would all be considered variants. Okay, anything that is not the most common, okay, is considered a variant. Okay, anything that's not a wild type would be considered a variant here. Okay, so one place where we do see multiple alleles is when it comes to our blood types again. Okay, so as we've seen already, the IA allele is going to code for the A protein or the A antigen on your red blood cells. Okay, the IB allele will code for the B protein on your red blood cells. Okay, but there's also a third allele. The allele is the little I allele. Okay, and the little I, okay, actually does not encode for any proteins. Okay, you don't get anything here, and so. These are essentially going to not code for anything in particular. So that's where we end up with the type O blood type. Okay. So while IA and IB are going to be co-dominant to each other, so they could both show up, like as we see with the type AB individuals. Okay. Both the IA and the IB alleles are both going to be dominant over the little I allele. Okay. So again, we have 3 alleles in the population. Okay. So the IA is going to be dominant over the little I. The IB is also going to be dominant over the little I. Okay? So that's going to be able to result in different genotypes that can give us different types of characteristics. Okay? So for example, if we have an IA allele that mates with another IA allele, okay, we're going to end up with the IA. We end up with type A blood. Okay? However, if we have an IA that joins up with a little I allele, okay, the A is still going to be dominant over the little I. Okay? And so we end up still having type A blood. Okay, we still have the A proteins showing up, okay, over the little I ones. Okay, same thing is going to be the case for the Bs. Okay, so if we have IB, interacts with another IB allele, here we have type B blood. Okay, however, if that IB also interacts with a little I allele, okay, then we have IB with a little I. Okay, the IB is still going to be dominant over the I, just regular old dominance. Okay, and so the B is going to be what shows up. So the B protein shows up here. Okay, now if you have an individual where you have a little I allele interacting with a little I allele, okay, that's where you end up with type O blood, where you have no antigens or no proteins on your red blood cell. Okay, so here we have more than just 2 alleles in the population. Here we're actually looking at 3 different alleles, the IA, IB, and the little I, that can result in different types of genotypes and resulting in different types of phenotypes as well. Okay, so that is multiple alleles here. All right, any questions on multiple alleles? Okay, cool. All right, so then the last one that we're going to be looking at here are sex-linked traits. Okay, so sex-linked traits is a type of inheritance pattern that specifically refers to genes on one of these sex chromosomes, okay? Either the X chromosome or the Y chromosome. Okay. Now, So far, all we've talked about were genes that are on the autosomes, okay? Anything that's not on the X or Y chromosomes, okay? So chromosomes 1 through 22, that's what we've been talking about so far, okay? However, there are traits that can be inherited specifically from the sex chromosomes, and most commonly it's going to be from the X chromosome, okay? So any gene that is found on the X chromosome is called an X-linked gene, okay? So it's only found in the X chromosome and not the Y chromosome, okay? Okay, so that's going to be what we're going to be mainly talking about when we're looking at sex-linked traits. Okay. However, there are some Y-linked genes here. They're not really as important as the X-linked ones. So we're going to just be focusing on X-linked traits. Okay. So when we're looking at sex-linked traits, the first place they were actually discovered is in fruit flies, in the Drosophila fruit flies. Okay. And so they actually have the same kind of sex chromosomes or sex chromosome patterns that humans do. Okay. Where females will have 2 X chromosomes, so an X and an X chromosome, okay, while males will have an X and a Y chromosome. Okay? So humans have an X and a Y chromosome for males, females are going to have an X and an X chromosome. And we're actually going to see the same trait happening, or the same kind of pattern happening in these fruit flies, okay, where females are also going to have 2 X chromosomes, males also have an X and a Y chromosome. Okay? Now the wild type eye color, okay, which just happened to be the dominant eye color, was red for these particular fruit flies, okay? And so the eye color is a trait that is found on the X chromosome for fruit flies. Okay, so they have so for example, the red eye color would have the XR allele, okay? X because it's on the X chromosome, and R because it's dominant, okay? And then the white eye color is gonna be X little r. Again, X because it's on the X chromosome, little r because it's recessive, okay? So again, the eye color is gonna be found on the X chromosome for fruit flies, okay? And so when we're looking at the females, okay, the females were going to still be considered either homozygous recessive or homozygous dominant. Okay, if they have 2 copies of the dominant allele, okay, they'd be XR, X big R, X big R for homozygous dominant, or homozygous recessive would be X little r, or they can be considered heterozygous, where they have X big R, X little r. Okay. Same kind of general trend we've seen already. Okay. However, in males, because we have 2 different chromosomes, okay, an X and a Y chromosome, we cannot be considered homozygous or heterozygous. Okay, we get a different term called hemizygous. Okay, so males are going to be hemizygous because we're going to have 1 allele on 1 X chromosome because we only have 1 X chromosome, okay, and then a Y chromosome. Okay, so we only have 2 combinations or 2 different types of genotypes that we can have. Okay, they can either be X big RY, okay, or X little ry. Okay, X because again we have 1 X chromosome. The R to signify whether it's dominant or recessive, and then a Y chromosome because males have the Y chromosome, while females do not. Okay, so that's that. Okay, so when we're looking at doing these different sex-linked traits and different crosses here, okay, we're going to end up seeing different results for where the recessive trait shows up in our offspring, okay, depending on where that recessive trait appeared in the parent generation. Okay. And so we'll do a few examples to kind of explain what I mean by that. Okay? But essentially, when we're looking at the offspring, okay, we're going to see different patterns of inheriting the recessive trait depending on where that recessive allele was found. Okay? So we're going to go ahead and do this example here. I've got a few examples of our X-linked traits. Okay? And so here we have a white-eyed male, and we cross it with a red-eyed homozygous female. Okay? So we're going to go ahead and do this Punnett square here. Here's our Punnett square. Oh dear, okay. All right, so if we have a white-eyed male, okay, again, the white eyes is recessive, okay? So what would the male's genotype be? Anyone have an idea? Right, it would be X little r y. Okay, so we'd have X little r here, and then the y would go on this side here. Okay, and then what about the female? If the female is red-eyed and homozygous, what would her genotype be? Right, X big R, X big R. Okay, so I'll go ahead and do her in pink here. So X big R, X big R. Okay. So again, we do our Punnett square just like we normally would here. Okay. And so in our first box, what would we end up with here? In our top left, we should have X big R, X little r, right? So X big R, X little r. Okay. What about this top right box? Right, we'd have X big RY. Perfect. Okay. All right, good job, Kimberly and Josie. Cool. And then third box, to the bottom left. Right, we'd have X big R, X little r. Okay. And then the bottom right, X big R Y. Perfect. So X big R Y. Cool. Okay. So a few things to kind of keep track of here before we start analyzing here. Okay. So one thing that you want to make sure we do is you want to make sure that each one has just 2 of the sex chromosomes. So you should have each box containing an X chromosome and a Y chromosome, or 2 X chromosomes. Okay. 2 sex chromosomes should be present in each box. Okay. That's number 1. Number 2, and this was a mistake that I saw when I was grading my lab's genetic practice problems, is that a lot of times people were looking at the males and assuming they were going to have 2 X chromosomes, okay, which is not the case. So males will always have 1 Y chromosome. They're going to have 1 X chromosome and 1 Y chromosome. Okay? And so those are 2 things to kind of keep track of. Make sure that when you're doing your Punnett squares, you have your females listed as having 2 X chromosomes, and then the male parent having an X and a Y chromosome. Okay, so make sure we keep those straight, because that was a mistake I was seeing quite a bit this week. Okay, so now that we've looked at our Punnett square, the next thing that we're going to do is we're going to go ahead and analyze our data here. Okay, so we're going to go ahead and skip the homozygous part, because I think we're okay with that, the homozygous and heterozygous part. But let's go ahead and just kind of analyze the phenotypes. So what's the probability of having offspring with red eyes? Looking at our Punnett square here, what's the chance of having offspring with red eyes? Yeah, so Kimberly's got this. It's 100%. Perfect. Okay, so 100%. All of them are going to have red eyes. Whoops. Perfect. And so the reason for that is because the dominant allele is going to be present in all 4 of these boxes. So all of the offspring here are going to have red eyes. Okay. Now, in addition to that, if you kind of extrapolate from that, the probability of having offspring with white eyes would be 0%. Okay. Perfect. Okay, so then now another thing that we want to be able to do here is we want to also be able to analyze what's happening based off of whether we have a female, whether we have a daughter, or whether we have a son. So in this case, I'll just kind of leave it at this, but this is, like I said, going to get a little bit more complicated when we go a little bit further here. Okay, so I'm going to go ahead and I'm going to show you what this would look like here. So yeah, so this is our Punnett square that we just came up with. Let me go ahead and clear this. Okay, and so as we see here, okay, we're going to have all of our offspring having red eyes regardless of their sex. Okay, okay. So let's do another scenario. Okay, let's say we have a red-eyed male, okay, and we mate him with a white-eyed homozygous female. Okay, so let's go ahead and do this Punnett square now. So again, I'm looking at, looking at our Punnett square. Hopefully the Punnett squares you guys are drawing at home look better than mine. So sorry about this. Okay, so let's go ahead and, yeah. Okay, so if we're looking for the red-eyed male, okay, what's the red-eyed male's genotype going to be? Okay, again, if red is dominant. X big R Y, exactly. So X big R Y, perfect. Okay, now if we have a white-eyed homozygous female, okay, what is her genotype going to be? Right, X little r, X little r. So I'll do this in green. X little r, X little r. And again, it doesn't matter which side you put the mom or dad on. Either way, it doesn't matter as long as you get the right answer. Okay, so now when we do our Punnett square here, let me do some. Okay. If we do this up Punnett square, so what should we end up with in this first top left box? Right, X big R, X little r. Cool. Okay. And then what about the top right? X little ry. Good. Okay. Bottom left. X big R, X little r. Okay. And then bottom right. X little ry. Good. Okay, good. Perfect. Okay. So now we'll go ahead and analyze our Punnett square. Okay. So what is the probability of our offspring with red eyes? Whoops, I made a mistake here. This should be. Perfect. Yeah, 50%, exactly. So 50% of our offspring would have red eyes. Perfect. Right, because we see 2 of our 4 boxes are going to have the dominant allele. Okay. Now, what's the probability of having offspring with white eyes? Right, another 50%. Good. Okay. So now let's take this a little further. Let's say we have a probability, or what's the probability of having a son with red eyes? Right, it would be 0%. Right, so this is where things get a little bit more tricky. So when we're looking at sons and daughters, you have to look at the offspring that have that particular genotype. Okay, so for example, if you're looking at a son, you only want to be looking at these 2 boxes here. Okay, okay, so then what's the probability of having a son with white eyes? %. Good. Okay. And then what about the probability of having a daughter with red eyes? Be 100%. Good. And then as having a daughter with white eyes would be 0%. Good. So that's what we would see here. So again, the key here, when you're looking at this kind of extra step here, is you want to look at the probability, okay, by looking at each individual offspring. So if you're looking at sons, okay, the sons are going to be the ones with the X and the Y chromosome, okay? So you want to look specifically at those ones when we're looking at the probability of having a trait within our sons. Okay? If we're looking at daughters only, okay, then we only want to be looking at these 2 boxes. Okay, so our answer is either going to be 0%, 50%, or 100%. Cool. All right, so that's what we would see there. Okay, so we were to do our Punnett square, let me go ahead and clear this out so you guys can see this a little easier here. Okay. So what we see here is that there will be red-eyed and white-eyed offspring. Okay. But if you have a female, okay, the female offspring, they are always going to have red eyes. Okay. All of the male offspring will always have white eyes. Okay. So I think we'll do one more example, and then we will go ahead and probably call it a day for today, and then we'll finish this up next week. Okay. So we'll do one last example here. So here we're looking at a white-eyed male, and we cross him with a red-eyed heterozygous female. Okay, so again, if we do our Punnett square, okay. I think that's the best square I've done so far. This is rad. Okay, so if we have a white-eyed male, what's his genotype going to be? Again, if white is recessive. X little r y, perfect. Okay, and then the female, red-eyed heterozygous female, whoops, what would her genotype be? X big R, X little r, right. X big R, X little r. Okay. Okay, so now if we were to do our Punnett square, what would we find in that first box? Right, we would find, what would we get here? X big R, yeah, X big R, X little r, perfect. Okay. What about this top right? X big RY, good. Okay. What about this bottom left? X little r, X little r. Okay. And then the bottom right, X little ry. Good. Okay. So now we go ahead and analyze our Punnett square here. So if we're looking at our total offspring, what's the chances that we have offspring with red eyes? Right, 50%. Okay, 50% because again, we have 2 of our 4 boxes containing that dominant red eye allele. Okay. What's the chance of us having an offspring with white eyes? Another 50%. Good. Right, because again, we have 2 with the with only the recessive allele. Okay. Okay. Now, if we're looking at a son with red eyes, what's the chance of us having a son with red eyes? Right, it'd be 50% because again, you're only looking at the sons which have the X and the Y chromosomes. Okay. So between this one here and this one here, okay, only 2 boxes. One of our 2 boxes has the red eye allele, which would be this guy up here. Okay. What's the chances of having a son with white eyes? Okay. Again, 50%, perfect. Okay. It's 50% because we have one of our 2 boxes, one of our 2 male boxes containing the white eye allele. Okay. What about the chances of having a daughter with red eyes? %, right? Because we have one of our 4 boxes containing our dominant allele. Okay. And then a daughter with white eyes would be what? Another 50%. Perfect. Because again, one of our 4 boxes contains both recessive alleles. Cool. So when we're doing these Punnett squares, when we're doing, especially when we're working with sex-linked traits, okay, we have an extra step that we didn't do previously. Okay. In sex-linked traits, what we want to do is we want to be able to look at the phenotype of the offspring as a whole. Okay, but then we also want to do an extra step in terms of determining what the probabilities are if we have a son and a probability of if we have a daughter for each individual trait. Okay? So we need to be able to do that extra step here. Okay? And so if we were to kind of do our Punnett square here, this is what we would come up with. And so we would see that we would have half of our male offspring having red eyes and half of the female offspring having red eyes. Okay? And so here we kind of see that depending on where the recessive allele shows up in the parent, okay, that's going to change what the offspring are going to look like. Okay? Here, half of the females and half of the males had red eyes. Previously, okay, here we saw that all the males would have white eyes and all the females had red eyes. And again, that's just because of where the recessive allele shows up. Okay? So it's always going to be a little bit different. Okay, okay, so we will leave it here. We'll pick this up again on Tuesday. And then, so if there's any questions, I'll go and just stop sharing now. Okay. Does anyone have any questions on anything we've talked about so far? Okay, so if not, I will upload the classwork, so that will be available for you guys to knock out today. I will work on getting the homework taken care of too. We didn't get through as much as I was hoping to, so I'll have to adjust the homework. But I'll put up the homework and that'll be up by later today. Okay? So make sure you complete the homework by next week, and that'll help you get a little bit more practice with these Punnett squares here. Cool. All right, so with that, you guys are good to go. If you have any questions, I'll be here for, oh, yeah, the extra credit. So sorry. Before you take off, let me kind of show you one quick little thing here. The extra credit, like I said, is up on Canvas. So is everyone able to see the Canvas page? Once this thing decides to load for me. So just as a quick little thing here, there is some extra credit on Canvas. And so, here we go. So if you go over to the Chapter 8 module, okay, is everyone able to see the Canvas page? Okay, so here on the Chapter 8 module, you're going to see at the bottom here, there's going to be an extra credit for simple dominance and simple recessiveness, and then the extra credit for the non-Mendelian genetics. You have everything you need to be able to do both of these. These will be due next week. Okay, so these will be due next Tuesday in class. Okay, or sorry, they'll be due on Canvas next week. So make sure those are uploaded. Turn those ones in when you get a chance. And so each of these is worth 10 points extra credit. So you have a chance to earn up to 20 points extra credit just by doing these. Okay? And depending on how many of them you get right. Okay? So make sure you complete that as well. That'll give you a little bit extra practice, because Punnett squares will be something that you will have to deal with on the next exam. Okay? There's going to be a lot of Punnett square questions. So you need to be very, very comfortable with that. Okay? So that is that. So I'll just leave it there. So if you have any questions, feel free to ask. If not, you're good to go.